That is indeed our title, Cancer and Evolution, Why We're Not in Kansas Anymore. And it's an interesting thing. It is now roughly 140 years since Alfred Wallace and Charles Darwin independently proposed the theory of evolution by natural selection. And in that time, there have been a great many opinion polls in the United States on the subject. It has never been true that more than 50% of Americans believed that this is the correct explanation for the origin of life on Earth and its subsequent diversification. And so it is a reasonable guess that in the classes that you teach, a significant fraction of the students will arrive from homes where this is either just not thought to be the right way of explaining things, or is thought in rather vigorous sense to be the wrong way of explaining what's going on. And so I thought it might be worth talking about it a little bit. And so this is a rough outline of what we're going to talk about. The first thing is, why does evolution matter? Well, it explains the diversity of life on this planet, in particular because natural selection rewards exceptions to rules, so that when organisms arise that have new ways of doing things, they can exploit resources that are not available to other organisms. And so it, in a sense, encourages the diversification of life. It explains host pathogens arms races, and at least in the case of HIV, which Rob was just talking to you about, it explains pathogen doctor arms races. So when people get HIV, when they become HIV positive, if the virus didn't mutate, and if you like, change its spots, the therapies that scientists and drug companies have developed, the things that inhibit the viral reverse transcriptase, that inhibit the protease molecules that help the virus make infectious particles, those drugs would be capable of either curing patients entirely of HIV or at least allowing long-term survival. And what happens, unfortunately, is that the virus mutates to become resistant to those drugs and essentially escapes from the therapies that doctors throw at it. And this is an example here. It's a pathogen, the host. It's not so much the human host, because we haven't dealt with HIV long enough to produce an evolutionary response, but it's at least the cultural response of medicine and pharmacology. It explains, in a sense, I've just dealt with this, why AIDS kills you. It's because the virus can evolve, that it can evade sophisticated therapies. And what I want to talk about a good deal today is that evolution explains why cancer kills you. So as I'm sure all of you know, cancer is caused when cells in your own bodies start to disobey the strict rules that determine where and when in your body cells grow and divide, and allow cells to escape from controls that normally regulate cell proliferation. Okay? So, but before I talk about, and so one of the reasons for talking about cancer is, if you're going to have the argument with people who say that, well, this doesn't seem like the right explanation for anything because it's just a theory, you can't watch it, it happens too slowly. In the course of HIV infection, in the course of cancer, in the course actually, in addition of the evolution of antibiotic resistance in bacteria, you can watch in a human lifetime evolution in action, and you can demonstrate that it is the ability of cancer cells to evolve that enables this disease to arise in the first place, and secondly, enables it to escape therapy. So one of the realizations in the last 15 years has been that cancer cells make genetic mistakes much more frequently than the rest of your cells. And those genetic mistakes in the forms of mutations, changes in the numbers of copies of chromosomes, enable cancer cells to evade our attempts to kill them through chemotherapy and radiation therapy in just the same way that mutations in HIV allow it to escape medical intervention. So a strong argument to students, or for students with their parents, or for you with students' parents, is that you can actually see evolution happen in these diseases. They are evolutionary diseases. In the case of AIDS, it's evolution of the virus that makes the disease so pernicious, but the virus could infect us at least initially without having to evolve within us. In the case of cancer, cancer is entirely an evolutionary disease. If mutations did not occur in our bodies, if those mutations did not give certain cells an advantage in terms of being able to replicate and grow faster, there would be no disease. Cancer is an evolutionary disease. Okay? So here are some of the objections and the rebuttals to those objections about evolution. So the most famous one is, it's just a theory. And so that's a claim made as if the rest of science were different. And the answer is it's not. Everything in science is a theory. You can never prove that anything is true in science. You can only disprove hypotheses. So some of you are probably thinking, well, that may be true for experimental science. It's not true for mathematics, because you learned when you were in high school or college, and some of you may teach students now, geometrical proofs for things. And the answer is, yes, that is just a theory too, because when you do geometry, or any branch of mathematics, there are axioms. An axiom is a statement like two parallel lines never meet. Okay? So I don't know how many of you have been several million light years to test that theorem. Okay? And it turns out that in mathematics there's a thing called the axiom of choice that is required for a lot of very sophisticated mathematics, way beyond high school or even college level mathematics. And within the mathematical community in the 20th century, there has been a very vigorous debate as to whether the axiom of choice is something that you should accept or not. And the balance of belief appears now to be that it should, but there are still dissenters. And so it's a way of reminding people that everything in science is a theory. Theories make predictions, you test the predictions. If the theory survives the predictions, you become more confident that that's the right explanation for how things work. But you can never prove it's the right explanation, because there could always be some prediction that you haven't yet tested. And when you test that prediction, you find that it's false. And when it's false, at least in principle, you're required to throw the theory out. At least if you're the average scientist, you don't throw it out, you modify it. And you keep modifying it until you decide it's so hopelessly broken you need a new theory. So the next problem with evolution is that maybe it denies God. So the best answer to that is that the founders of population genetics, the study of how evolution actually happens in population, a field that developed in the 1920s and 30s, of the 5 people who founded that theory, one of them was a practicing Anglican lay priest, another one was deeply religious Unitarian, and so on and so forth. There's no fundamental conflict between believing in evolution by natural selection and believing, for example, that God created the universe, or indeed that God created the world, or even potentially that God created the first living thing. Because what evolution really deals with is what happened after the first living thing existed. And so it's only a problem if God made species one by one and she made them 6,000 years ago. And if that's what you believe, yes, there is some fundamental inconsistency between your view of creation and evolution, but there are equally fundamental inconsistencies between your view of evolution and the accepted view of cosmology, or indeed the geological history of the Earth. So the next point of view is that evolution incites amorality, okay, in the particular sense that if all we're doing is struggling to leave more descendants to reproductive age than our neighbors, almost any sort of bad behavior is justifiable. That you can steal, kill, rape, pillage because you're just trying to fulfill your biological destiny. And the same thing is true only if all of science does. The idea that what goes on when you see and recognize things is that chemical signals cross between one nerve cell to the other, and that those chemical signals cause electrical impulses to run along nerves, could be used to say, well, actually there is no such thing really as free will, because this is all just electrical transmission in your brain. This is in fact an illusion of free will. And to be honest, I actually believe that. right? It's a very powerful illusion. It's actually an illusion that's necessary for human society to work. It's necessary for me to continue to feel like I'm making a productive contribution, okay? And like lots of things, one is capable of believing two apparently inconsistent things at the same time. And the argument that it's the sole province of evolution within science to possibly act, if people wished it to, as a justification for amoral or immoral behavior seems like it's a weak argument. Okay, so this, I've already talked a little bit about this. Is there any evidence for evolution? Yes, and I won't talk about this today, but if you look at sequences of the same gene in different organisms, so we have hemoglobin that carries oxygen from our blood to the rest of our bodies. So do monkeys, so do mice, so do fish, so do chickens, so do frogs, so do birds. And if you sequence the hemoglobin genes from all those different organisms, you can use the differences in their sequences to infer who's more related to who. So there's a cultural analogy to this. I actually have a movie, but we'd need sound, so we'll skip it. The cultural analogy is that there was a time in human history where there was no printing, okay? And most books existed in monasteries, and when you needed to make a new copy, you copied the book, okay? Now, the scribes were monks, and although they might have been absent many of the temptations that happened to people out in the real world, they probably still got sleepy from time to time, and for sure they made mistakes. And when they copied and they made a mistake and they took the book back to their home monastery, someone who came from the monastery down the road to copy their book was going to copy the mistake, right? And so on. And so you can actually trace the spread of information through the Dark Ages by looking at the surviving copies of those books and looking at the pattern of the mistakes, because you can actually tell where the mistakes were first made. And you can see who went to those monasteries to copy their books and who copied the mistaken copies and so on and so forth. And the way that DNA sequences encode history is exactly the same, okay? And the last argument, so one of the things that's hard often for biologists to understand is why is it that evolution of all scientific theories gets such a bad rap, right? The Big Bang, in some sense, is playing even closer to God's home turf, right? But you don't see the institution for cosmic creation or intelligent design of the universe. And no one seems to be protesting too vigorously against Isaac Newton. And so one of the questions is, why is it that evolution causes so much trouble? And a friend of mine who's a scientist who comes from the Midwest and grew up in a small and very religious town, her assertion is that it sounds like evolution is aiming for perfection, and that perfection in many different religions is the province of God. That we're imperfect. If you're Catholic, you're born into mortal sin, etc., and so on. And the only person who's allowed to make things perfect is God. And if evolution is so progressive and is so hell-bent on making things perfect, that's trespassing on God's territory, and that's bad, and we better slap it down. And so one of the things that I want to tell you is that evolution is not necessarily a progressive force. It is a blind force, okay? For example, the evolution of HIV in patients who are receiving various different sorts of therapy may be helping the virus. It is certainly not helping people. But you can at least argue in that situation, well, it may not be perfecting people, it's perfecting the virus. Cancer is the perfect example, because in cancer, the evolution of cells in your body is remarkably short-sighted, because not only is it going to kill your body, it is going to kill the cells that are evolving themselves. And so I use that as a way of saying that unless we define perfection as being the closeness of your resemblance to James Dean, now I know that there are probably some of your students of the female persuasion who might define that as being close to perfection. But that's in the Rebels Without a Cause sense, but that's the only way that cancer gets close to perfection. So here's the roadmap of the lecture. I'm going to introduce you to cancer. I'm going to talk about the distinction between what's called your germline and your soma. And we're going to talk a little bit about multicellularity and the division of labor. We're going to talk about mutations in your germ cells and your somatic cells. So your germ cells are your eggs in your sperm, and all the cells that were required to, all the cells that are in the cell lineage that connects them back to the fertilized egg that you developed from. And every other cell in your body is your soma, and all the somatic cells, when they put you in that deal box, or me, and put us in the ground, all those other cells are going to die. The only cells that will survive into the future are those lucky eggs and sperm that get fertilized to make children. And they, in turn, can go on. Their descendants can go on. And so that's why we talk about this distinction. We're going to talk about cell proliferation and cancer. We're going to talk about how, in normal cells, you have to balance cell birth and cell death. We're going to talk about multiple changes in cell behavior as being the causative agent of cancer. And last but not least, and depending on how fast we go, we'll talk a little bit about the epidemiology of cancer. That is the science of deciding who gets cancer and why. And we'll talk about cancer resulting from multiple mutations. We'll talk about environmental contributions to cancer. And we'll talk about genetic contributions to cancer. Okay. So I'm going to define some terms first. Lots of times biologists talk about cell growth when they mean one of two things, and sometimes both. The increase in the size of cells and the increase in number of cells. I will try and be more precise and use cell growth to refer to the increase in the mass of cells, that is, cells getting bigger. Excuse me. And cell proliferation to refer to increases in cell numbers, that is, cells becoming more numerous. A tumor is a group of cells that has grown and proliferated inappropriately. A benign tumor is a tumor that is confined to one part of your body and whose cells do not escape. And if you have a benign tumor, a surgeon can operate on you and remove the tumor, and basically the threat of cancer is gone. If you have a malignant tumor, cells can escape, migrate, and settle at new locations, and once they're there, they can establish secondary tumors, a process that I'm sure most of you have heard of called metastasis. And once tumors have metastasized, the surgeons are by and large out of luck because the chance of finding all those secondary tumors is small, and that's when you start giving people chemotherapy and radiation therapy, and these are blunt and not fully effective tools.
I have a question.
Yes.
Benign tumors, what I understand, should be taken care of early on. I don't know if all, but I know that in some cases they should be removed because they eventually will become malignant, or they have the potential.
Absolutely.
Now, is that a good example of evolution?
Yes.
Benign malignant?
Yeah. So it's cells in the tumor acquiring new properties, and those new properties allowing them to go new places and proliferate and divide and escape from the surgeon. Okay? Yes, it is a good example. And so the dictionary definition is that cancer, the term cancer, is applied only to malignant tumors. Okay? Now, the way the word's used on the street and in your classrooms, that's not necessarily true because people tend to lump these two things together. They say it's benign cancer, it's malignant cancer, but really this is cancer. Okay? So actually now I realize, how easy would it be to do sound?
There's a sound wire right there.
Okay? Perfect. Sound wire.
It's right on the top.
It's at the top.
Small. Oh, it is. Okay, great. All right. So I'm going to illustrate two different challenges set by two U.S. presidents.
I believe that this nation should commit itself to achieving the goal before this decade is out of landing a man on the moon and returning him safely to the earth. No single space project in this period will be more impressive to mankind or more important for the long-range exploration of space.
Okay, quite the Boston accent, didn't he? So this is a speech made in 1961. 8 years later, Apollo 11 landed on the moon, and the cost in 2005 dollars was about $135 billion. Okay, enough to buy the Red Sox about 250 times over. Okay, so here is another president, Richard Milhous Nixon, in 1971 during the State of the Union address.
I will also ask for an appropriation of an extra $100 million to launch an intensive campaign to find a cure for cancer. And I will ask later for whatever additional funds can effectively be used. The time has come in America when the same kind of concentrated effort that split the atom and took man to the moon should be turned toward conquering this dread disease.
Okay. So the question is, what's happened since then? So the easy thing to answer is, how much does it cost us? It cost about $200 billion between the federal government, money raised through things like ACS, contributions of pharmaceutical companies to research. And so the question is, have we cured cancer? And the answer is, well, it's a bit like the war in Iraq. You can declare victory, but it doesn't necessarily stop people from dying. And it's an interesting question to ask. Actually, let me show you the numbers first. So here I'm going to show you the data for two different cancers. So these are data from the United Kingdom. It's for men. These are deaths per 100,000 men annually. This is a disease called Hodgkin's lymphoma. Lymphomas are tumors of your immune system where your white blood cells become malignant. In the case of lymphomas, they grow at a single point in your body. So these cells aren't circulating in your blood the way lots of white blood cells are. And it's in distinction to leukemias, which are similar cancers, except now the white blood cells circulate around your body, and in some senses that makes them harder to cure. This is a particular sort of lymphoma. It's called Hodgkin's lymphoma. And what you'll see is the rate at which people have been dying has gone down roughly 4 times. So this is one of the success stories. There are now much better therapies than there were 25 years ago for curing Hodgkin's lymphoma. And lots of people, including Harvard's former president, Larry Summers, have been successfully cured of this disease. Unfortunately, this is a different cancer. This is colon cancer. And there are 2 things you should notice. The first is this line is not going down very much at all. We have about maybe a 15% reduction in the death rate. And the second thing is that the scale here is different. This starts at 30 deaths per 100,000 men, whereas this one started closer to 2. So about 15 times as many people are dying of this disease, and not that much has happened in the period since Nixon declared war on cancer. And the same picture is true for the other big killers. The other big killers are lung cancer, prostate cancer, and breast cancer. And so it's an interesting question to ask, why is it that we can cure, why is it we can send people to the moon and we can't cure cancer? And the answer is that sending people to the moon is an engineering challenge. The science that was required to send people to the moon was basically developed by Isaac Newton. Okay? And if you understand Newtonian physics, you can figure out how fast a spacecraft would have to go to escape from the Earth's orbit. And then it's merely, and I use merely not to denigrate engineers, but at least if you're a scientist, it's merely an engineering challenge to find out how to build rockets that are big enough and fast enough so that you can reach that escape velocity, and build navigational systems that are precise enough so that you can actually get them not just out of Earth's orbit, but land them on the moon and get them back again. That's a ferocious challenge, but it's a technological challenge, not a scientific one. The challenge with cancer is to understand how it is that the cells in your body that cause cancer differ from the rest of your cells. And until you understand that difference in some detail, you don't know what the engineering challenge is that you have to meet in order to kill those cells. Okay? So I've mentioned these things before: multicellularity, germline mutations affect your children, because those are the cells, the eggs and sperm, that are going to be your children. Mutations in somatic cells affect you, and the only mutations that you have to worry about are the ones that could give you cancer. So here is an illustration of the distinction between germline and soma. This is a family that you may have met before. So this is an egg produced by Queen Elizabeth. This is a sperm produced by Prince Philip. This is the fertilized egg that is going to be Prince Charles. Okay? Divides once, twice, and a bunch more times until there's a little peanut here that contains about 3,000 cells. At this stage in human development, you can start to identify what will become the germ cells, the cells that are going to go on and become the cells in your testes, if you're a man, excuse me, that are going to produce sperm, and this is the British royal line of succession right here. Okay? And all the other cells, the one that give rise to the big ears and the hair and the brain that makes pronouncements about British architecture and the state of the world and so on and so forth, all those cells are the soma, and all those cells are destined to perish when Prince Charles dies. Okay? The germline contribution comes in the egg, and this donor, Princess Diana, is already dead. Okay? And here we just do the same thing again. Fertilized egg, 2 cells, 4 cells, peanut. British line of succession, potentially one of your students' names here. Okay? Prince William, soma, cute, charming, attractive bachelor. And this is designed to illustrate it's only these cells that matter. And in fact, all any of us are, sadly, from an evolutionary point of view, is we are projectiles for our eggs and sperm, because our eggs and sperm are the things that carry our genes and have the chance of making them immortal. And in fact, a large part of the behavior that you probably see every day in your classrooms, at least if you teach at schools which have both male and female students, reflect the ability of that evolutionary drive to control the behavior of adolescents and young adults. I think you know what I mean. Okay? So that's the distinction. And germline mutations can cause cancer in our children. So there are certain inherited cancer-prone conditions, and in that case, the mutations get passed you through your germline, and if you have children, you pass them on to your children. Somatic mutations can cause cancer in us. Okay? And so we're actually going to worry primarily about these sorts of mutations because they account for the vast majority of cancer. And so we're going to worry now about how many mutations your students will pass on to their children. And so we'll do this by worrying. We'll start with an egg here, and we'll worry about what happens to the DNA that was in that egg, which is one set of 23 chromosomes, the 23 you got from your mom, between the time this egg got fertilized and the time a woman produces the egg that will give rise to her children. Okay? For most of the time in between, they're not just the 23 chromosomes, because you got 23 from mom and 23 from dad, but when you form this egg, you'll go through meiosis and you'll go back down to 23. And so there are about 25 cell divisions that separate this egg, the one that got fertilized, from this egg, the one that a woman produces every month, and if fertilized, can develop into a child. And so we're going to ask how many mutations occur in the intervening period. You can ignore HIV. This comes from a slide from the class. So you have about the egg that you started with had about 3 billion base pairs. The rate at which you make mutations at a rate of about once in a billion. So if you multiply these numbers together, each time a cell divides, there are about 3 new mutations. Since it takes 25 cell divisions to make a new generation of human beings, to go from the egg you started with, if you're a woman, to the egg you'll produce to produce the next generation, you get to multiply this number by 25. So now you multiply mutations per base pair per human generation times the 3 billion base pairs of DNA that you're going to pass on to your children, and you get to 75 mutations. So that when you had children, if you did, or when your students have children, each child will contain 75 brand new mutations that their mother bequeathed them. And the numbers are actually slightly worse if you're a guy. It's more on the order of 100 to 125. Now, it turns out to be the case that 95% of your DNA is just junk. Okay? So only about 5% of those mutations are going to end up in DNA that matters. So roughly speaking, you get about 4 mutations like that from mom and about 6 mutations like that from dad. So every newborn infant has about 4 plus 6 equals 10 mutations that their parents didn't have. And the question is, why doesn't that kill us, right? If roughly half of those mutations are bad, deleterious mutations, why aren't most infants born dead? And the answer is something that probably many of you teach your students. It's the difference between dominant and recessive forms of a gene, okay? So when you look at people who have blue eyes and have brown eyes, they differ in a particular gene that is responsible for eye color, and technically we say there are different alleles of that gene, one of which is responsible for your eyes being blue, the other of which is responsible for your eyes being brown. And again, as I suspect many of you know, and probably quite a lot of you teach your students, in this case, the blue allele is recessive and the brown one is dominant. So if you get a brown copy from mom and a brown copy from pop, your eyes are brown. If you get a brown copy from mom or pop and the other copy is blue, your eyes are still brown because brown is dominant to blue. And it's only true if you get a blue copy from both parents that your eyes are blue. And it turns out that most of the mutations that we were just worrying about, those 10 new mutations that children get, are recessive. And as long for most of the genes in your body, you get a good copy from either your mom or your dad. Why are they mostly recessive? That's a good question. Because mostly they make the genes, they make the proteins less capable of doing things, okay? And so for most of the genes in your body, half as much of the protein responsible is enough, okay? So that, for example, if you imagine having a mutation in a digestive enzyme, like trypsin, the enzyme that's in your stomach that digests food, and you only got a good copy from one parent, having only half as much trypsin still enables you to digest food, and there are other enzymes in your small intestine, and you know, for the vast majority of genes, half is enough, okay? There are genes where that's not true, okay? And well, so actually sickle cell anemia is a good case, okay? So if you are a heterozygote for sickle cell anemia, you're basically fine, okay? You have to have both mutant copies for something to go wrong, okay?
They can still pick up if you're
You can detect people as heterozygotes, but the reason you detect whether they're heterozygotes is if 2 heterozygotes get married, you need to test all the embryos so that you can tell people they're going to have a child who has sickle cell anemia. But they are basically healthy people. Yes?
Actually, I have a question. If you have one good gene and one non-functional gene, do you really have half the enzyme, or are there feedback mechanisms that you wind up making a lot more?
That's an excellent question, too. So you might think that evolution had been really smart and this went on enough that there should be some mechanism so that you go, oops, I only got half as much, better make some more from the good copy, okay? It turns out that in bacteria, that's sort of what's called autogenous regulation, the ability of proteins to regulate their own synthesis, is quite common. Even in the humble eukaryote that my lab works on, our friend the brewer's yeast, and definitely in us, that is the exception rather than the rule. And so most of the time, half the number of good genes means half as much protein. Okay? Sometimes as a result of that, you trigger other mechanisms, right? And for some things like growth factors, like erythropoietin, the thing that makes you make more red blood cells, and you illegally take if you want to win the Tour de France or the marathon, if you had only one copy of the erythropoietin gene, you would make less red blood cells. And as a result of making less red blood cells, you would be stimulated to express the one good copy more. And that's an example where through a very indirect feedback loop, you would make more of the protein. Great question. Okay, so I want to emphasize that your bodies, I guess the next thing I should say is, An interesting question, if you point out this distinction between the germline and the soma to your students, that you can point out to them, is it means that your body is this enormous, incredibly complicated cellular cooperative. There are at least 100 different types of cells in your body. There are at least 60 trillion cells total. This is a cooperative to get your eggs and sperm to where they need to go. And however proud we are of our own or our societies or our students' accomplishments, again, from the brutal evolutionary perspective, our bodies are the chauffeurs for our eggs and sperm. And an interesting question you can ask them is, why is it that the somatic cell should be so happy doing all the work, chauffeuring the eggs and sperm around, when they're going to go to the cold, cold grave, and eggs and sperm, at least the lucky ones, get to live forever? Okay? And the answer to that question is that the chauffeurs have exactly the same genes in them as the eggs and sperm. So their genes are being immortalized too. And the direct demonstration for that is our friend Dolly the sheep. Right? You guys remember Dolly the sheep? So the way Dolly was made is you take L sheep egg and you suck out, after it's been fertilized, you suck out the nucleus that contains the information from mom and pop. So now sheep egg destined to have not much happen to it, except the scientists put back into it the nucleus from a differentiated adult cell. And because that contains exactly the same genes, you still get a sheep. It walks like a sheep, it barks like a sheep, it must be a sheep. Okay? And that's the direct demonstration that it doesn't tell you that that's why these guys are quite happy to be the chauffeurs, but it does tell you that they have the same genes, and therefore you can make the logical argument that chauffeurdom should be just fine. So here are some of the different sorts of chauffeurs. This is a neutrophil, this is a white blood cell. These are the guys that run around in your blood and track down bacteria, eat them, and oxidize them to death. They live about 3 days, which is not very long. There are about 20 billion of them in your body, and as a result, because they don't live very long, you have to make 80,000 of them every second. Okay, so this is a way of driving home how much cell division is going on in your body. These are red blood cells. These live longer, but there are even more of them. There are about 20 trillion red blood cells in you and your student's body. So 2 million of them are being born a second. So if you teach a class that lasts about an hour, 2 million are being born a second. In an hour class, about 9 billion of these cells will have been born. Okay, that's 1.5 times the population of the Earth. That's per student. You can multiply by the number of students in the classroom. Okay? And this is a contrast. This is a scale bar. This is 5 microns, 5/1000th of a millimeter. So these cells are bigger, smaller. But these cells are ginormous because this is 100 microns here. These are Purkinje cells. These are neurons in your cerebellum, the back part of your brain that's responsible for the fact that I can stand up straight. And these cells, this is the body of the cell. This is a huge projection. This is what's called a dendritic tree. It's where this nerve cell gets all its inputs, and it integrates all that information. It sends an electrical signal out through this thing here, which is the axon. These cells live as long as you do because they're born actually before you are. And there are only about 20 million of them. So this is an example, this is a rose gallery of a small fraction of the hundreds or so different sorts of cell types that make up your body. And so what we're going to keep your body in the right shape and in the right function, you have to regulate the birth and death of all those different cell types, and you have to get them to play nicely together. It's sort of like the perfect socialist state, from each according to their abilities, to each according to their needs. And so we're going to talk about balancing birth and death, and we'll talk about multiple changes in cell behavior causing cancer. So I want you to think about what would happen if cell birth and cell death were not completely balanced with each other. So we're going to imagine a type of cell that divides every 24 hours. And we're going to say that the cells are born 1%, 1.01 times faster than they die. And we're going to use what seems like a bizarre analogy at first, because what relevance does a tennis court have to your body? So again, some of you probably teach to your students that the area of your small intestine, if you open it up and got it perfectly flat, is about the equivalent of a tennis court, and it's that area that enables you to absorb things from your food effectively. So this is our tennis court. And so you go, well, 1% off, how bad can that be? So here's a month of being 1% off, since these cells are dividing roughly once a day. So this is 1.01 to the power 31, which turns out to be 1.36. So a tennis ball's gotten a little bigger. It's easier to avoid the double fault. It's harder to lob your opponent, or I guess it's easier to lob your opponent successfully too. Okay? You go, well, you know, it's 36% bigger, not such a big problem. Here's a year. This is 1.01 to the 365th, so the tennis court is now 38 times bigger in area, and it might be a little hard to fit this all in. Okay? So this is an example that you have to have some way of very precisely balancing supply and demand. The invisible hand of Adam Smith must descend over your body and balance supply and demand. And I'm going to give you an example of one of the cases where we think we understand a little bit about how that works, and that is erythropoietin, which I mentioned before. So erythropoietin is a growth factor which stimulates the proliferation of the cells that end up giving rise to red blood cells. Erythropoietin is made by cells in your kidney, and it is made in inverse proportion to the amount of oxygen that comes by your kidneys. Less oxygen, more erythropoietin. So if, for example, you were to take your students on a skiing holiday for the Christmas holidays to Colorado and live at 10,000 feet in the Rockies, the skiing might be balanced by yet another month with your students. What would happen is everyone would feel a bit weird the first day, because all of a sudden you're at altitude, there's less oxygen in the air, you have to breathe harder, you actually deliver less oxygen to your blood, and your kidneys would be getting less oxygen, and they would make more erythropoietin. And eventually, over a period of a few weeks, the actual number of red blood cells in you and your students' blood would go up, and it would go up because the cells were being born faster than they died, because your body can detect there's not enough oxygen. When you go back to sea level, there's it's not quite there's too much oxygen getting to the kidneys, but there's enough getting there so that the level of erythropoietin goes down, and now for a while you give rise to red blood cells, you birth red blood cells more slowly than they die, and so your red blood cell count will go back down again to the level that's appropriate at sea level. Okay, so that's Adam Smith and the red blood cells. Okay, so the next thing I want to say to you is that lots of the time, again, this may be something you teach students about the exponential proliferation of cells. You may even in some high school classes grow bacteria and watch them increase logarithmically in number. So 1, 2, 4, 8, 16, so on and so forth. And I'm going to show you an example to point out that most of the cells in your body are not growing and proliferating, and they certainly aren't doing it exponentially. And here's why that is impossible. This is a blow up of the W in God We Trust on a penny. And this is the dimensions of a single cell, roughly 10 microns in diameter. So now we're going to let that cell divide every 24 hours, and its progeny do likewise, just like this. After a month, we have a solid sphere of cells about the size of the penny. So there's the W in God We Trust. After 2 months, solid sphere diameter of the famous tennis court. 3 months, you can go, if Cambridge is in the middle, Walden Pond is about here, and poor old Henry David Thoreau is deciding that life is not so simple as it used to be. Okay? And if you wait, to term of human pregnancy, you would have a solid sphere of cells about 10,000 light years in diameter, which is about the size, is a galaxy about 1/10 the size of the Milky Way. Okay? So exponential cell proliferation better be rare. So now I'm going to give you some big scary numbers. The number of cells in your body is about 60 trillion. The number of cell divisions in a lifetime has too many zeros to write on the slide, so it's about 10 to the 16th. So you can ask, how many times, and then the next thing to tell you is the mutation rate is about once in a billion per base pair per cell division. So let's multiply this number by this number. So any given mutation will have occurred in your body 10 to the minus 9 times 10 to the 16 times, which is 10 million times. So every individual possible single base pair mutation in your DNA, by the time you've died, will have happened 10 million times. So now, if a single mutation could give you cancer, you would have had cancer 10 million times in your lifetime. So that clearly can't be right. So I'm going to explain all the different things that have to go wrong to cause cancer. So this is a normal human cell. It obeys incredibly strict rules about where and when to divide, rather as such a prim and proper Victorian young lady might. These cells divide only when told to, only when they see growth factors like erythropoietin. There are different growth factors for different sorts of cells. They would die rather than misbehaving. Victorian shame has been taken to ridiculous extremes. They stay close to home. Most cells live and die in a very small area of your body. That's obviously, there are exceptions. Your red blood cells go whiz round and round and round. And in particular, they die if they stray far from home, and they're extremely careful with their chromosomes. The frequency of mutation is very low, and cells do a very good job of segregating their chromosomes so that when one cell divides, its daughters each have exactly 46 chromosomes. And it takes about 5 mutations to turn this figure into this one. Okay? So this is the original rebel without a cause. Disobeys all the rules, divides at will. Bad behavior does not induce the sense of molecular shame required to cause death, wanders throughout the body, and is extremely careless with its chromosomes. If any of you guys want any of these slides, you just have to send, I'll give them to Tara and you can send an email and you can get them. If you do, you should ask your students how many people can identify both these figures. Who's this? Emily Dickinson. So this is a little harder. Sid Vicious, very good. That would be harder for your students than for you because, like me, some of you are of the era where you might have listened to the music that the Sex Pistols made. Okay, so you do much better than Harvard undergraduates. Harvard undergraduates have quite a lot of trouble with this one. Okay, so this is a slightly better or more precise list of the genetic changes. Cells grow and proliferate under conditions where normal cells do neither. They ignore signals that tell badly behaved cells to kill themselves. This is the process called apoptosis or programmed cell death. They induce the growth of new blood vessels. So if a tumor arises and the cells start to proliferate, the tumor will grow until it's a few millimeters in diameter, and then the cells in the middle have a problem. They are a long way from a blood vessel, and it takes a long time for oxygen and glucose and amino acids to diffuse into the middle, and most of them get eaten by cells they have to go past. And likewise, it's hard to get rid of things like CO2 and urea. And so there's an effective limit to the size a cell can grow without blood vessels growing into the tumor. And so one of the sorts of mutations that tumors acquire and are selected for, because as soon as you acquire these mutations, the tumor can grow bigger, is the ability to induce the growth of new blood vessels, so they literally grow into the tumor. Then, to become malignant, they have, again, as someone pointed out, as part of an evolutionary process, they have to acquire mutations that allow them to enter and exit blood vessels and form tumors at new sites. And last but not least, this is now starting to require a lot of mutations. The estimates are a typical tumor cell has somewhere between 5 and 10 mutations that are required for its behavior. To accumulate all of these in a row, it's been appreciated over the last 10 or 15 years, requires a mutation that doesn't change the growth property of the cells, but changes the rate at which cells make mutations. Okay? Because the faster cells make mutations, the faster you can accumulate all these things. Okay? It's a if you like, this is a selection for mutations that make evolution faster. Right. So this is a reminder for me to tell you that cancer is an evolutionary disease. I already said this. Evolution leads to imperfection, selecting for increased growth and proliferation. These are cells, at least in that chauffeur analogy, they're like, we don't need no damn germ cells. We don't need no chauffeur. We're going to live forever. We're just going to proliferate like crazy. And the answer is wrong, okay? With the exception of a really scary thing called transmissible venereal canine cancer, which is a cancer where the cells can actually be transmitted from dog to dog. So these guys really figured out how to do without the chauffeur. And a few genetic changes can drastically alter cell behavior, so the idea that evolution is too slow to have ever possibly happened certainly isn't true for cancer. And prolonged strong selection selects for mutations that increase the mutation rate, things that scientists call mutators, and that's that. So when were you guys expecting to finish?
Actually about an hour from when you started, so about
So in about 5 to 10 minutes.
It's up to your schedule too.
Yeah, but I guess, let me tell you what's ahead. So I can talk about statistical evidence that cancer results from multiple mutations. Environmental contributions to cancer and genetic contributions to cancer. It will take more than 10 minutes. So what I'm going to do is I'm going to pick one of these things and talk about it, and then I'll stop and take questions from you guys, okay? And I think it might be worth talking about this because one of the questions that obviously arises is why do we get cancer in the first place? Okay? This is such a horrible disease. Why hasn't evolution selected to make us resistant to cancer? And the answer actually is that it has. We have all sorts of genes that are required to keep us from getting cancer. And one of the forms of genetically inherited cancer is mutations in those genes. Okay? So they're what are called guardians of the genome, for example. They're genes that try and keep the mutation rate low, and they have probably been evolved precisely to keep us from getting cancer. But why do so many people get cancer? And the answer is actually remarkably simple. This is the incidence of cases of cancer per 100,000 US women. So this is newly detected cancer per year at different ages, starting at age 0 and going up to age 80. Okay? So the first thing you can see is that although there are tragic examples where cancer affects children or young adults, this is the tiny, tiny majority of all cancer. Okay? And this arrow is here because this is what life expectancy was in the United Kingdom in 1750. And at life expectancy then, there weren't too many people. Okay, so life expectancy 30 doesn't mean everyone died at 30 because an awful lot of people died here, but it meant that not that many people got out here. Okay? And there were a lot of other things to worry about, like infectious disease and starvation. The second thing to point out was not only were people dying relatively young, but once you've stopped having children, you're dead as far as evolution is concerned, right? Because we're just the chauffeurs for the genes. Once the genes have been driven to their destination, the chauffeur can be unemployed, okay? And menopause, which is, in late 40s, early 50s now, was certainly a lot younger then because people were much less well nourished and so on and so forth. So the answer is there has been selection against cancer. It's just that we've unfortunately figured out how to live long enough to get cancer. And the one thing I'll close by telling you about is how you can use curves like this. So when I said that we know roughly how many mutations it takes to get cancer, that has required, there are two approaches. The one that I was talking about before was cell biologists and molecular biologists looking at cell lines derived from cancer patients and with great pain and trouble tracking down the differences in cellular behavior and ultimately the genetic differences in single genes that underlay those behaviors. That's literally probably tens of thousands of people years worth of work collectively. So I'm going to tell you about a shortcut. It won't tell you what the mutations are, but it will tell you how many you need. And it comes from looking very carefully at this curve and is an interesting topic to discuss with more mathematically minded students, and as a way of reminding students who aren't more mathematically interested that if they're interested in biology, it's good to know something about math. Okay? So we're going to use an analogy. We're going to use a dice analogy. This is going to be a weird game. The dice has 100 sides. You only get to throw it once a year. Okay? and winning here is actually losing. Winning is getting cancer. So if you only have to get one mutation to get cancer, you need to throw 100 once to win, that is lose. If you need 2 mutations, you need to throw 100 on each dice, but you don't have to do it in the same year. You can sort of keep one in the bank. So you can ask, well, what's the probability of winning each year? So the probability of winning in the first year is 1% if it's just one dice. Throw it once, 1% chance of rolling 100. Chance of winning, again, winning really means losing here, sometime in the second year, sometime by the time you finish 2 years is 2%, because you're going to have 2 throws of the dice, 3 years, 3%, so on. If you've got to throw 100 on each dice, it's 1% of 1% in the first year, 2% of 2% in the second by the time you've done 2 years, and 3% of 3% by the time you finish 3 years. So there are 2 things you can see instantly. These numbers are a lot smaller, so that's good, less likely to get cancer. But now the rate, instead of going up linearly, is going up as the square of the number of years. So here, that's plotted on a graph. So this is for 1 dice, 2 dice, 3 dice, 4 dice. Okay, and you can't really see the sort of the curvature here because the numbers are so low. Okay, and once it's 3 dice and 4 dice, you can't even see that anyone's losing down here. So now we're going to plot this on a scale where we use the log of the chance of losing and the log of time. And lo and behold, and you can convince yourself doing relatively simple math that this will work out, these lines are all straight. Okay, and they're all visible on the same graph. And what you can see importantly is that they have different slopes. And the slopes tell you how many dice you have to roll. And a British epidemiologist called Richard Peto figured out that this is what you needed to do to figure out how many mutations you needed to get cancer, because you just take the age-specific incidence, you plot it on a graph like that. So we'll just go back. Boom, boom, boom. You just take this graph, you plot the log of this against the log of that, and it's not linear, but there's a period where it's starting to go up where it will be linear. And you get a graph like this, and it turns out it's somewhere between the line for 4 and the line for 6. Okay? And so this is the epidemiologist's evidence, which nicely corroborates the molecular biologist, that you need somewhere between 4 and 6 independent mutations to accumulate sequentially to give you cancer. So what I think I will do there, rather than rushing through any more stuff, is stop and take questions. The cells that make milk in your breast, the cells in your prostate, all the big cancers, they come from the division of epithelial cells. Those cells, it's less clear that the right way of thinking about those populations is there's some ridiculously small number of stem cells that account for everyone. So that, for example, in your large and small intestine, there are these things called microvilli, or sorry, actually called villi, not microvilli. And they go up and down like that. They look like little fingers. The cells that ultimately provide all the cells, the stem cells, lie at the bottom of the crypt. The argument is there are 2 or 3 per crypt. Okay? It is those cells in which the mutations probably have to occur in order to give you cancer. The other possibility, and stem cells are sort of hot and sexy, so they're scientific waves of fashion. So a lot of people want to think it's just stem cells. In principle, you can have mutations that occur so that the cells that the stem cells give rise to that normally would have just divided a fixed number of times now are capable of indefinite division. And for some diseases like leukemia, the idea is that the initial mutations occur in a stem cell. And that gives rise to some very low-grade disease that it's hard for clinicians to pick up. Eventually, the last mutations, the ones that give you the leukemia that's going to kill you, occur in the descendants of those cells. And so that those cells now act themselves like stem cells, so they keep dividing indefinitely. Okay? Does that help?
What about cancers of nerve cells and striated muscle cells?
So cancers of muscle cells are rare, okay? Basically because there's not all that much cell division that goes on. And so there are 2 sorts of neurological tumors. There are things called neuroblastomas. Those are cells that were supposed to become nerve cells, neurons that kept dividing. Those mostly affect children because in adults there is very little, if any, cell division that's making nerve cells. The things that kill grown-ups when they have brain tumors are what are called glioblastomas, and they're the glial cells that support the nerves, and they continue to divide through our adult lives, and mutations accumulate in them and can give rise to cancer. We work on brewer's yeast, so I know cancer mostly secondhand, and I talk to people who work on cancer. Clinically, I take your word for it if that's what clinicians say, but if so, I don't know why. I mean, the worst sort of cancer, I mean, there's a simple answer. The worst sort of cancers are the ones that you can't kill by radiation therapy and chemotherapy, and in principle, it's the ones that are the most genetically unstable because they accumulate new mutations that enable them to become resistant to the drugs that you're throwing at them, right? And what makes in the pathway of mutations that give rise to a cancer, some cells have higher rates of genetic instability than others is not well understood. It reminds me to tell you something that's important. Another reason that cancer is so hard to cure is it is not a single disease, right? Different cancers arise from different sorts of cells. Even from the same sort of cell, there are different pathways of mutations. There's more, for each of those different sorts of mutations I listed, there's far more than one, there are far more than one gene where mutations can occur and give rise to a particular defect, like the ability to escape programmed cell death. And what that means is that drugs that will kill one cancer can be completely ineffective against another. So there's a disease called chronic myelogenous leukemia, which is a particular sort of leukemia. There is a miracle drug for CML called Gleevec, which totally specifically inhibits the proliferation of these cells. It is essentially completely ineffective against almost every other form of cancer. And so one of the big questions about cancer therapy is what in the long run is going to be more helpful? Is it going to be having a designer drug like that for every different sort of cancer? Or is it going to be having better versions of the blunt, kill every dividing cell in the body version of cancer therapy that has been used until very recently? So most of the drugs that people take, like Taxol for chemotherapy, they just do a number on dividing cells. And the reason chemotherapy and radiation therapy makes people so horrendously sick is because basically you're just laying out the dividing cells. You're not particularly specifically attacking cancer cells, and lots of the medicine that goes into designing chemotherapy and radiation therapy is essentially trial and error. I don't think people know well enough. I can tell you that you can get different forms of colon cancer where you get different sorts of mutations. So there are two inherited diseases where if you are unlucky, in genetic diseases where if you are unlucky enough to inherit them, you are essentially certain to get colon cancer, okay? So one of them is called HNPCC, which stands for hereditary non-polyposis colon cancer, okay? And that's a disease where one of the systems in your body that checks after you have copied your DNA and roots out any mistakes, something that's called the mismatch repair system, is defective. And you make mistakes in DNA replication about 100 times faster than regular folk do. And if you have that disease, you get colon cancer. And in that colon cancer, you make what are called point mutations, changes of an A to a G or a C or a T, 100 times more frequently than regular cells do. There's another disease which is called APC, which stands for adenomatous polyposis coli. Again, it's a hereditary disease. If you get it, you're essentially certain to get colon cancer by your early 40s or 50s. In that case, it's a disease, the mutations, amongst other things, affect the pattern of chromosome segregation. So if you look at the cells from people who have that form of colon cancer, basically every different cell in the cancer has a different number of chromosomes. Whereas in HNPCC, all the cells have 46 chromosomes. It's just the sequence of the DNA isn't right anymore. Okay? So those are two different, completely different ways that cells can become genetically unstable, okay, as examples. I mean, and I'm sure it's the sort of question that students ask, like, what's the worst, what's the best, can you rank all these things? And the answer is, for most of them, scientists don't know enough to be able to say, okay, this has a higher mutation rate than this. We can say qualitatively, those two different sorts of cancer have mutations in completely qualitatively different ways.
Are all organisms capable of becoming cancerous? Like you studying yeast, you see yeast?
No, because yeast, so the difference is that yeast aren't multicellular cooperatives,
So you
right?
have to be multicellular to be cancer?
Yeah, basically, right? So in a yeast cell, one cell is an organism, right? So
Right.
there's no tragedy of the commons. There are no rebels without causes.
But
They're
I
just
mean, you don't see this wild yeast cell that all of a sudden is taking off and dividing.
no,
too
because they're
much.
just growing as fast as they can.
Anyway.
Anyways, right? And so the question is, if you look in multicellular organisms, do you see cancer in everything? And the answer is, it's sort of yes and no. So if you look at mice and rats and frogs, yes, absolutely. Stuff clinically indistinguishable from us. And in fact, mice have been incredibly useful tools because you can make mutations, the equivalent mutations in mice, and look and see how they get cancer. And it's been a very powerful way of understanding the genesis of cancer. If you look at things like nematode worms and fruit flies, which are very popular things for scientists to use to try and understand basic processes in biology, do they get cancer in the sense that we do in rats and mice and frogs and birds? No. Can you get mutations that cause uncontrolled cell proliferation? Mm-hmm. Yes, but those organisms live such short times that the chance of the mutations arising in the lifetime of individuals is essentially zero. And they actually don't. First of all, they don't live very long, so there are less cell divisions, and they also don't have that many cells altogether. So in your lifetime, I said there are 10 to the 16 cell divisions. In a worm's lifetime, a nematode worm, there are 1,000. Okay? So you just don't have to worry about it that much. But you can get mutations, if you're a scientist, germline mutations, if you look hard enough, that produce uncontrolled cell proliferation that looks like the first stages of cancer. But is it a problem in the natural history of these organisms? No.
It's like those big tortoises that live over 100 years.
Yeah, that's a good question. It's like in very long-lived things like that, is cancer a problem? Now, the thing is, well, it's an interesting question what the distribution of their lifespans is in nature. If it's like UK life expectancy in the middle of the 18th century, yeah, then probably the big ones are getting cancer. I don't know. That's an excellent question. No idea what the answer is.
Trees. I'm sorry.
Well, no, so the difference is in plants. So plants get things called galls. You've probably seen galls. Those are actually due to bacteria introducing genes into the plant cells that make the plant cells proliferate more or less uncontrollably. The difference is that plants, although they have a circulatory system for getting water and sugar around, it doesn't move cells around, so there's no way you can have metastasis in a plant. So effectively it means that those galls, which are in effect plant tumors, have to be benign, exactly. Yes?
What about comparison between something like warts or moles or those types of growths to two different types of cancer?
Well, warts are basically on their way to being benign tumors, okay? And it's just that, and for example, so there are things called genital warts, particularly in women, that are caused by what are called human papillomaviruses. Some of those, having had those, is a very strong predisposing event towards uterine cancer. Okay, so it sort of makes the relationship clear. The average wart on your skin that you go to Walgreens and buy burn wart off, chance of that becoming malignant is incredibly small, and it's usually annoyed you and you've gotten rid of it way, way, way beforehand. But wart is basically a very benign tumor.
Thank you.
Thank you.